So now we are going to see what are the important interactions and the forces involved in the drug receptor complex. So in this slide, there is a model molecule which is present that is depicting wide range of interactions with the receptor pocket. So one is covalent bonding, ionic or electrostatic interaction, ion dipole or dipole-dipole interaction, hydrogen bonding, charge transfer interactions, hydrophobic interactions. This we have already seen, like just like a waxy layer which are present and sliding down inside it, and halogen bonding and van der Waals interactions. These are all the weak forces. One by one, we will let us see. These are all the very important to provide the drug action. Not one interaction or one force which is not going to resulting into a pharmacological action and a drug long acting nature. Many forces at the same time they will be working in the receptor, and that is producing and what the desired effect we wanted to expect it to get without toxicity. We don't have any expectations with the drug receptor complex a permanent bonding. That is not a requirement. So it should have an association also, dissociation also. So the first bond, which is very strong among all, is a covalent bond. You know, in the organic chemistry, you have already learnt. In the covalent bonding, they are the strongest bonding generally formed by the sharing the electron between the two atoms. That is the reason it is the strongest bond. And majority of the drug combine with the receptor by the weak molecular interactions only, not like by the covalent bond, because we don't expect any irreversible bond. These interactions like covalent bonds provide a strong link between drug and receptor, but individually the interactions are reversible. Even though they form, then only it is possible to have a drug feasibility. It formed by the drug receptor interaction except with the enzymes and the DNA. If it is happening like that, it is irreversibly damaged or mutated. That is the reason covalent bonding we need to handle very carefully. The second interaction is ionic and electrostatic interaction. Most of the times our biological pH of the plasma where our drug is traveling is having pH of 7.4. Where we are having a drugs with which are weakly acidic and weakly basic in nature 99% of times. So the basic groups what are present in the interaction site are amino acids chains of arginine, lysine. They are protonated. That is, therefore they provide the cationic environment because they are having a NH2 primary amine, and protonated they will be having a tertiary amine. They will be providing a cationic environment. Whereas the second one is acidic groups such as carboxylic acid, which are present in the amino acid like aspartic acid and glutamic acid. They are deprotonated to give the anionic groups. So these two are the interaction sites where it is having an opportunity to form as a ionic bonding. So the drug receptor groups will be mutually attracted because of the opposite charges provided by the opposite charges. So this ionic interaction can be effective at distance further than those required for the other type of interactions, because just like we are having a magnetic metal and magnet, even in the long distance they get attracted opposite charges in the similar manner. So the North Pole and South Pole, where it is having a same charges, they repel, and opposite charges they attract. So they persist longer. It means we can predict this type of interactions are influencing our induced fit. That is one thing which you are going to see. So this figure, if you see the electronic interactions, so the wavy lines present the receptor cavity where we have got a dotted lines which are providing the charge, and we are having a plus and minus. So these two opposite charges providing the ionic or electrostatic interaction. The third one is ion dipole and dipole-dipole interaction. These are all things which you have already learned in physical pharmaceutics. So you are well versed with it, but in this context, we need to see with a drug and receptor, both what are the sites or the functional groups involved in that you need to aim at. So because of this ion dipole and dipole interaction, the greater electronegativity of the atoms such as oxygen and nitrogen, sulfur, halogen relative to that of the carbon. So these bonds in the drugs and receptors, where the X is the electronegative atom, will have the asymmetric distribution of the electron and produces the electronic dipole. These dipole will be acting one side as a one charge positive, other side it will have a negative opposite charge. So that is the reason we call it as a dipole. So the dipole can be induced by one ion, that is ion induced dipole, and another dipole, it will induce another dipole adjacent to it. So this will happen because of this asymmetric distribution of the electronic configuration in the atoms that will provide the charge. These dipoles in the drug molecule can be attracted by the ions, similar as that of the electrostatic interaction what we have seen, but here it is the little bit weaker than them. So this we are going to have an ion dipole interaction. By another dipole, so one dipole is getting attracted with another dipole, then we call it as a dipole-dipole interaction in the receptor, and provide the charges of the opposite sign are properly aligned and they will be getting a bonding. Because of the charge of the dipole is less than that of the ion, what we have seen in the electrostatic interaction and ion dipole, a dipole-dipole interaction is weaker when compared to your ion dipole and electrostatic. Dipole-dipole interaction is weaker. The fourth one, very important in our drug design, is hydrogen bond. So hydrogen bonds are the type of dipole-dipole interaction formed between the protein of electronegative atom, which is containing one hydrogen, and one or more electronegative atoms which are already present for the pairing with the non-bonded electrons. So the most significant hydrogen bond occur molecule where the electronegative atom are nitrogen and oxygen. We can call them as a X and Y. We can name any way we can to designate. X removes the electronic density from the hydrogen, so it has a partial positive charge and which is strongly attracted to the non-bonded electrons of the Y. So the interaction is denoted by X, each is having electronegating atom, which is containing one hydrogen, that is getting a hydrogen bond with another electronegative atom, which is present adjacent to it. So this can be possible. And you need to remember here, we have to see that this will be happening with the intermolecular or intramolecular also. Sometime it will be having a intramolecular hydrogen bonding interaction that improves the lipophilicity. That's what we have yesterday seen. So this X referred as the hydrogen bond donor, where we are having a H plus, and Y is the hydrogen bond acceptor. Y is not containing in this context hydrogen atom in it. So that is the reason these have the electronegativity and the degree of ionization. The proton can be shared between the two groups. They will be having depending upon the hydrogen presence along with them. Charge transfer complexes. This is the fifth type of interaction we are going to have with a force. When a molecule or a group that is a good electronic donor comes in contact with molecule or a group that is electronic acceptor, so donor may transfer some of the charge to the acceptor. This forms the charge transfer complex. This will be happening only when they are coming to the proximity with each other with a distance, with a critical distance, they are going to have a bonding. So the donor group containing electrons, they are like alkenes and alkynes and aromatic moieties with electronic donating substituents and the groups. So that contain a pair of non-bonded electrons such as oxygen, nitrogen, sulfur, and the acceptor groups contain the electron deficient orbitals. So these are all also called such as alkenes, alkynes, and aromatic moieties. It means in both the cases we are having the same type of functional groups. They are behaving, so they can act as acceptors also, donors also, and we withdraw the substituent and weakly acidic protons. These groups are receptors that can act as an electronic donor, such as the aromatic ring of tyrosine, which is present containing aromatic ring in the amino acid, and carboxyl group of the aspartate, they will show such type of force. The sixth is the halogen bonding. This is, we are going to optimization part. We are going to use this bond as a critical one. So it has well established that a covalently bonded halogen atom can act as an electron acceptor, Lewis acid, to undergo the halogen bonding with an electron rich donor atom, such as O, that is oxygen, nitrogen, or sulfur. On the basis of the crystal structure, the quantum mechanics and molecular mechanics, this part of quantum mechanics we will be separately dealing in the preferably 5th unit at the end. So at this particular time, we are not going to deep into that. Just by first we see the forces now, the energy is involved. Lest we go later onwards, we go to this part. It was found that many of the halogens, like you got a chlorine, fluorine, bromine, so these are all iodine, the halogens to the oxygen or nitrogen bond distances were equal to or less than the sum of the respective van der Waals radii, indicating the formation of the halogen bonding. So they are having, when coming to the proximity, they will tend to form a bond similar to the hydrogen bond, but it is also a weaker bond. So not like a covalent and ionic bond. So the strength of these interactions is in the order of, so to form, iodine is having greater than bromine, bromine is greater than chlorine, chlorine is greater than fluorine. The fluorine is having the lead interaction, that is having a strength to form an interaction to have a hydrogen bond. Another force what it is very important is van der Waals or London dispersion forces. So atoms in the non-polar in nature may be temporarily non-symmetrical distribution of the electronic density, which results in the generation of the temporary dipole. So this dipole is intrinsically present with the asymmetry or it is induced by the adjacent atom. So as the atom from the different molecules, such as the drug or receptor, approach each other, the temporary dipoles, because we are using, it is not a permanent dipole. The permanent dipole, like we are having ions, which we are already seen. So it is a temporary. When the molecule is having a dynamics, this dipole will also alter, and the charge distribution will also taking place as a change. So because of that reason, we are using computer softwares and generating the scores, because we are not taking a drug molecule as a rigid or a complex which is formed with a receptor as a rigid, because we are making so many alignments and we are seeing which alignment changes these dipoles and making a charge distribution and gives free energy. We are going to calculate. This is what the alteration in the direction or the shape and the movement of the drug that will have an impact on your bonding. So one molecule induce the opposite dipoles in the approaching molecule. This will cause the interaction. So consequently, intermolecular interaction known as the van der Waals forces resulting in and causes the binding to the drug to the receptor. These weak universal forces only become significant when these are in close surface contact of the atom. Otherwise, it is not possible, not like a magnetic metal and magnet, what we have seen in the ionic bonding. It is not like that. When they are coming into the proximity only, they will be having activated and they will be forming as a bond. Now, once this charge, what we have seen, is having an influence on your receptor bond, drug receptor bonding, there is one more important is the cavity size, because the size matters. Unless otherwise the induced fit is there, if the molecule is bigger than the receptor pocket, that is cavity, then it will not enter. So this is not the case with induced fit, but that is a different story. And in most of the cases, the size is going to be very much important. So binding site and molecular interactions, we need to see the cavity size prediction also. So the binding site provide the base for the interaction of the two molecule and describe the ability of the receptor to form bond with another substances, which are either the endogenous ligands or we are giving a ligand in the form of a drug. It is either agonist or partial agonist. Based on the bound molecule, the binding site can be a protein or protein nucleic acid or protein carbohydrate or protein lipid or protein small molecule binding sites. So this is actually, you see, the protein is macromolecule. The site can have to accommodate the another protein also in its pocket because of the tertiary structure of it is having a tertiary structure and the pocket is created. Here we have got a recent advancement like AlphaFold. So this part we will see little bit later. This AlphaFold has made this artificial intelligence to predict drug tertiary structure without doing our NMR and axonocrystallography. So this AlphaFold, it is having a greater change in our research perspective. It has shifted in a different manner, and we can predict with the computer software because of this particular predictions. What we are already made is working well. So these protein and all which we are having is the big molecules are also getting accommodated. Similarly, molecules which are having less than 500 molecular weight also getting accommodated in the pockets. So the binding of the drug molecule to the plasma proteins, that is albumin and lipoprotein and globulins, is the major determinant of a drug distribution. If these interactions are not present, most of the times the drug will be excreted very fast, and they will be leaving our body through urine because they are free in nature. Glomerular filtration will be present, and they will be going out of the body, and they will be having a very short half-lives. And this is not good also. So we need to have the binding of our drug molecules with the proteins. That is very much critical to have a optimum persistence in the body. So the binding site prediction based on the role of protein, the binding site can be categorized into active and regulatory sites, active site and regulatory site. So the active site of the protein binds to the ligand molecule and performs the enzymatic activity that will produce your pharmacological action. Whereas the regulatory site binds to the regulatory molecule, which either activates or inhibits the process of the binding at the active site of the protein. So it is tend to have an antagonistic and partial agonistic. So these binding sites determine the strength and the type of interaction between the protein and ligand molecule. But means both regulatory and active sites both are very much critical. So to predict the protein binding sites, most machine learning based approaches are used because we are having these active sites which can be predicted by the AlphaFold, and what the pocket is exposed to have, which amino acid is exposed, and what is its functional group, we can easily find out, and we can predict what is the corresponding group in the molecule of a drug which is going to have an interaction. So these machine learning models are developed using the structural sequence and evolutionary information that is actually helping us to predict a drug which is a drug or a promising fragment from the database, chemical databases. This one we will be seeing little bit later, chemometrics. So this one, what we are going to learn, that is how they are going to get a predict in the machine learning. We will see little bit later. Now we are going to see that this machine learning could be possible because of these sites, which what we are predicting is really present in the most of the molecule, which is already been structure known by the crystallographic methods. So the predictions and what is really present both are matching. So the protein complexes modeling and docking software are the few examples of using this evolutionary and structural information. Because of these predictions are working, we are able to make a softwares for the docking studies, and we are going to have your fragment-based drug design by selecting a correct moiety, which is going to have a better interaction. So the evolutionary algorithm of a binding site prediction utilizes the information of the multiple sequence alignment and assumes that binding site residues are conserved for the evolutionary related proteins. This is what we are having in a growing approach and linking approach of a fragment-based drug design. Because of this evolutionary algorithm, it will suggest what type of the fragment can fit into that particular interaction site and regulatory site. This is what software is doing and helping us. Whereas the energy-based methods calculate the interaction potential between the protein and the ligand, where we have already seen comparative force field analysis. So this is what the interaction potential between the protein and the ligand, and utilizes the 3D structural information of the protein and protein ligand complexes from the protein databases. So we are going to see what type of this is very much important for the repurposing of our old drug into the new condition by placing the old drug into the new protein and see how the interaction is present, whether can we use in that particular treatment also. That can be used. So binding site prediction methods are based on the evolutionary algorithms and energy-based algorithms and geometric algorithms. The geometry algorithm is also very much important because the pocket is not even. It is having a up and downs, peaks and valleys, and it is having irregular in shape. And ultimately, the charge loci is going to work for this particular geometric metric. And we are going to use the geometric algorithm also. So one is taking a size into the account, another is taking the energy into the account, so another is having a shape into the account. So these three together, we are going to design a drug molecule. So how this particular binding site prediction tool we are going to have is depicted in the diagram here in the flow sheet. You can write by just seeing the data in it. Protein ligand binding site databases are the binding days collected from the literature, which is already existing passed in the databases. They prepare the data set for the binding sites predictions, which are having a similarity that energy simulations what we are going to use. So extracting the sequence based on the features such as similarity and conservation score. Based on the score, they will be odd thing out. They make it and select the best fit into the account and they select. So the energy-based features such as the probe interaction site and contact potentials and geometry-based features, they will be considered to picking up the best molecules, and which are having the top scores, they will be segregated and they will be taken into the further consideration. Computational methods and optimization algorithms for developing a binding site prediction tools using the sequence and the energy and geometric based features and finalize which moiety among the what we have selected is going to fit in the pocket. So the prediction and validation of the binding site using these tools, it will help us to design a drug without any failure. And if we go to the lab also, it is going to work in nearby future. That is what we are doing in the CAD. We want to fail the molecule in the first so that we will be avoiding the wastages. So what are the prediction tools and servers which are available? So these are several computational tools for the pocket and cavity predictions have been developed. So these are all not like a cavity which we are seeing with X-ray crystallography. This is used by the computational tools by using a computer software, which is based on the evolutionary algorithm, energy-based algorithm, and geometric algorithms. So each software, they have got their own advantages and disadvantages and cost factors also. And freemiumware and freeware and premium software are also available. According to the budget, we can select them and go with our selections. Some of the other approaches have been developed which combines 2 or more methods to improve the accuracy of the cavity or the pocket predictions. It is an extensions which we can use it further confirmation and confidence gaining. So the binding site predictions use different parameters and method for prediction have a different accuracy because each having their own limitations. That is the reason we cannot expect one is going to solve all the problems. So where we are predicting that there is a issue or there may be some lacuna, we have to use another software or another approach to avoid that. So these are all what you have seen here, the drug, how the software and look at the drug molecule, which is fitting into the this spirals inside the cavity. Important to consider following points in the de novo drug design because of we have got three things. One is a charge, another one is a size, another one is a geometry, shape. That is the reason we cannot go simply. Some points we need to consider before taking a decision in the de novo drug design because we are starting from the scratch. This is very, very important slide. So you have to see that these mistakes should not be occurring when we are doing a de novo drug design. Flexible molecules are better than the rigid molecules. This is the first thing we need to consider. Whenever we are taking the dipole-dipole interaction, that will be changing with the distance present between the interaction site and the molecule. So when we rotate our drug molecule inside the pocket in the 360 angle, so the distance will alter and charges and force fields will also alter. So we cannot make the drug as a rigid, and when it is present in one direction, whatever we are thinking that is only present in the socket. We cannot have such a rigid molecular structure. So we need to consider a flexible molecule. That is one thing we need to keep into the mind in a de novo drug design. Whether it is a fragment-based drug design or we are going to use any another approach, this is a fundamental we need to keep in the mind. So because these flexible molecules, they are likely to find the alternative binding conformation. Should they fail to bind as expected in case of rotations, we need to confirm. If the rigid molecule fails to bind as predicted, it may not bind at all. That is the reason we can reject in the early stages. Another point is that it is pointless to design the molecule which are difficult or impossible to synthesize. It is working very well. Scores are very good. And with respect to geometry and charge, that is force and your size, everything is perfect. But that molecule, what we have seen is impossible to synthesize because it is a peculiar in nature and it costs very high and it is not feasible like appearing. So that is no point in proceed further. The third point is, it is pointless to design a molecule which need to adopt unstable conformation in order to bind. So we are having a conformation. When it is binding in that conformation, it will work as a drug. But that conformation is not stable because we cannot expect like a rigid molecule. It is a flexible in nature. That is the reason these unstable conformations we cannot go ahead as a drug molecule further. That is the reason we need to consider. The fourth point we need to consider is there are some different structure between the receptor time from the different species. Like we are taking a similar insulin receptor of a rat or a guinea pig or human or monkey. So these racial differences and species differences will be present. Even in the human being also, we have got so many polymorphism in the enzymes. So some races will have a poor acylator, some are having a fast acylator. So this racial differences are also present. But when we are making a new drug, it is at least we need to consider species differences. So when we are considering a protein of a rat, we cannot expect the same thing protein of a human, which will be working, which we are going to launch in the product, not like a veterinary medicine. So this is very much critical. The fifth point is that we are going to have the structure of the binding site used for the de novo site is based on the protein that is not human in origin, we should never consider for the drug design. So we have already seen that species differences are present. That is the reason. So the protein which is not a human in origin, we should never take for the pocket size determination and force analysis, and we cannot go ahead. Ultimately, why we are going to have this is, even though you get a success, there is no guarantee that success will be retaining in the human beings. So these points which we need to consider in the de novo drug design, including your fragment-based drug design, so that you will not have a final failures, but it is having a failure we are knowing at the first itself. We are avoiding. This is prevention is better than cure. So we have to prevent such a type of situations in our design so that we will not have a failure in nearby future. Now coming to the softwares used in the de novo drug design. So site point connection methods. So we are having a linkers where we have already seen in the fragmentation design, LUDI is used. And fragment connection methods, we have got a splice, new lead, proligand, they are all present. Sequential buildup, where we are starting with one atom and growing in an evolutionary manner. So there we are going to use legend, grow and sprout. Random connections and the disconnection methods, which we are going to find a short path methods, where we have seen one is a short path method. The short path methods where we can use the concepts, MCD, DNLG, the softwares are present, they can be used. So the software at present, we are having as a list at the end of after completion of 5 units, you will be able to know in chemistry based and biological based softwares, and we are having a both together we are going to have and docking softwares. So many softwares are going to come into the picture, then slowly we can memorize. This is only what the memory part in your Bloom's taxonomy level 1. This type of programs we need to just memorize it. If you are generally using in the practice these softwares, you can understand and easily what the software is having a ability and what is its limitation. You can easily understand, but at present you can just memorize it just like a Bloom taxonomy level 1. Now we have to compare de novo drug design, traditional methods, that is what we are using classically in age-old phenomena, versus AI-driven de novo design, where what we have got a computers now, we have got advances in the generative artificial intelligence. So what is the comparison? Because you should be able to know what is the recent trends, and old trends are outdated. Can we say like that? It is not like that. We need to have an understanding of both. And sometimes the classical and the traditional approach is also going to solve some problems. So that is the reason we need to see the comparison. Both are having some advantages and disadvantages we need to understand. So what you see in the Sprout and LUDI, these are all the traditional softwares we are using, where artificial intelligence and machine learning was not there. They are the programs rule-based program, like Lipinski rule of 5 and Weber's rules and what we have seen. These are all the rule-based and they will give the scores. Whereas the modern artificial intelligence platform, we have got a chemistry 4.2. So this is actually the advanced machine learning model, and it is having deep generative learning and it give the predictions. So like what we are having previously, the predictions which are only rule based, now we are predictions, we are going out of box. That is the advantage in these particular models. And here you need to understand little bit artificial intelligence. That part we will see in the 5th unit. Once our quantum mechanics, which we are going to touch there, how these artificial intelligence will help where the human being is failing. We will come to know how these we are going to have these generative adversarial neural networks. So got the hidden layers are present. What is the convoluted neural networks? These are all things we will little bit see later. That is actually a programming part and software build up part. So that we need to have a little bit understand, but we need not have a master in the artificial intelligence. But we should be able to know some terminology and what the workflow in that particular one we need to know. So the core method in the case of traditional is rule-based, and whereas the AI platform is a generative deep learning. Primary strength is highly interpretable, follows the strict geometric constraints of the binding pocket. And in the case of AI, it explores the massive chemical space, identifies the non-obvious and novel scaffold also. Previously, we never thought that this can be possible. That can be made into a possible in this, because it is beyond our imagination. These machine learning tools will provide us some solutions. A novel scaffolds and novel leads and hits it will generate. Primary weakness in both these softwares are limited chemistry diversity, because we are having a limited, because it is not trained previously. Only rules are being trained, and the rules based only it is present, often generate the molecule that difficult to synthesize. It can occasionally hallucinate. It means go from the nearness to the reality, go out to hallucination. That is in the AI terminology. Chemically impossible structures are also it is giving. So in the previous case of traditional, it is difficult to synthesize, but it is possible because we are using this traditional software. We are intentionally making some of the functional groups which is fitting to the rules. That is the reason easy to synthesize, but little bit difficulty is present. But in this case, it is impossible structures also it will predict by the computers, because it will hallucinate, it will go beyond your reality. So that is what is a misinterpretation, which will give, and it will have a bias also. There is one problem. So we'll see a little bit later in the AI tools, what we are going to learn in the 5th unit. Speed. In the speed, it is slow. The traditional methods are the slow, require the significant more manual oversight for the fragment selection. Whereas the AI models are ultra fast, because it is having a multiple hidden layers. They are trained in such a way that they select, deselect, code, decode very fast, and they can design these thousands of the viable candidates in hours and days, and we can pick up from the Pareto principle based on the scores very easily. Scoring. So release on the physics based, that is empirical scoring functions, where it is we can check in the laboratory and the mathematical phenomena with a logic, we can do it. But it is having a multi-objective and reinforcement learnings, where we are going to have creative algorithms which will provide the new reiterations, and it will bring the best solution to balance the affinity, toxicity, and solubility. That is the reason it is having a multi-objective. In the case of scoring, we may get the scoring of affinity and drug-like moiety is good, but toxicity is also good, great score is present. Then we can drop that moiety. In this, we have got a balance in the affinity, toxicity, and solubility, so that we will not have a problem in the admit also. Synthetic feasibility, often it is poor. It requires the chemist to manually fix the design for the synthetic routes. Here in the AI driven, often it is integrated, and automatic retrosynthesis are suggested by the computer itself by having a which path we can get the moiety easily. That is the end product easily from the what are the starting products are also it will suggest. Best use case, when the protein structure is very well defined, requires the specific interactions, then it is Ludi and Sprout like traditional softwares are good. Rapid discovery and difficult to drug targets, where we are not having anything known, and we want to have that, then the speed is critical. Then it is a artificial intelligence model of a modern age are going to be working better. So if you see a small case study where we are going to have a marine product, which is taken from the natural sources, databases. And these marine products, what we are taking is the least explored when compared to your herbal products. What we are already seeing, most of the data is present, and we want to explore as a de novo drug design. We pick up from the database of marine database, so that we will have a drug-like moiety from that database with guarantee to patent our moiety. So then we will be having a drug like likeliness and toxicophore presence, and these filters, it will give some moieties that will be working with a least toxicity and good efficacy. Then we do the molecular docking, where we are going to use a software gold and Autodeck Vina. So this Autodeck Vina and these softwares, it will provide the scores so that we can pick up from the moieties what are the best. Then we go to molecular dynamic simulation analysis, where we have got RMSD, RMSF, ROG, B factor, hydrogen bonds, RDF. So these are all analysis of the factors. We can further do the molecular dynamics when the molecule is moving. What is the dipole-dipole changes, and what is its impact on the scores, and which score going to be very much critical to proceed further, and that we can predict. Then we go to binding free energy calculations, where the Gibbs free energy should be very low. So for that, we are going to use MMG, BSA, water swap. This is also very much important. Sometimes we need to remove the water moiety and find the interaction. Sometimes we need to have a water effect also. Both are required to be seen. So we are going to find out in the presence of the water, without water. That is also available for the free energy calculations. Because when we are having a hydrophobic sites more, water effect is going to be less. When you are having a polar species, water effect we need to accommodate. Otherwise, it is not possible to have a good predictions. So this is what is critical at this particular point. Then we are going to have alanine scanning and mutant creation and re-estimation of the binding energies. And ultimately, when we are having with respect to the toxicity and your efficacy, ultimately at the end, we need to do adsorption, distribution, metabolism, elimination, that is excretion and toxicology analysis. So where we are going to use a drug likeliness, pharmacokinetics, medicinal chemistry, and toxicity into the account. And we are going to finally end up with a new drug, which is not already existing. This is what the one case we can see as a sequence, so that we will start with some database, which is a chemical database and which is containing only marine products, which are not acting as a drugs. Now we are picking up and modifying it, changing the functional groups coming out of a good drug, which is meeting all our requirement. Now I will open the floor for some questions. If you don't have any question, just I will ask myself a question and proceed further. 33 members are on board. So let us proceed. What are some questions which are generally present in the de novo drug design? How do you ensure AI designed molecule can actually be synthesized in the lab? Some question you may be getting in the mind, sir, we have made with a computer, it is not possible, or how we do it in the lab? That is actually what your question may be. Generally, this is a classic synthesis accessibility problem. There are certain combinations of the functional groups and pathways. It is impossible to make it in our synthetic lab. So the modern pipeline uses the retrosynthesis AI. There is a retrosynthesis AI platform. These tools break up the original product, that is a drug, into a basic fragments, and it will give the suggest the commercially available starting material and your reagents and your solvents, and it can make it the chemical reactions feasibility or not, it will also give. It means the computer is suggesting out the drug structure, and also it is giving a pathway to synthesize it also. If the AI can't find the path to make it, the molecule is discarded before it is ever reaches the chemistry desk. This is the one we need to do. It means even computer brain is accepting what are the possible reagents available in the world and what it taking, and it is considering all the databases. It is not having a feasible synthesis path. So it is beyond our human brain to think and synthesize it. It is better to drop at that time itself. The second question you are going to having in the mind may be that, is de novo design better than the virtual screening? In the virtual screening, what we are going to have that whether it is a high throughput screening or virtual screening, we are going to have a combinations of the product and testing them. And we are picking up the passing of all our criteria and we are taking it. So they serve the different purpose. Virtual screening is faster for finding the low hangout fruit. It is known library that we can easily pick up and go further. So it is in our reach, we can pick up. In that case, we can use the virtual screening. But it is not always the cases. We are having new diseases and we don't have any nothing we know about that particular disease. We want to proceed with the synthesis of a drug. Then it is very difficult to use these low hanging out fruit concept by using a known library. So we have to explore all libraries, and by the de novo synthesis and de novo design superior when you have the novel target where existing libraries are failed. We want to have a generative and predictive AI where we are going to get a new molecule. So when you need to bypass the existing patents, even though we synthesize, it is already falling under the similar hit or it is a lead. We cannot patent it. All our money is going to waste. We will not earn the money because the original patenter, it will sue us. That is the reason. In those cases, de novo is better, not the virtual screening, because virtual screening almost all are explored. We cannot find anything new. Creating an entirely new molecule scaffold is possible only in the de novo synthesis. What are the recent advances? Because you should be able to know what we are having in the textbook also you won't find. And there are people, they see regularly internet and find what is the one and the recent what the Nobel Prize holders, what they have done. And these are all the things which is a little bit we need to have an update with current knowledge. This is obviously required when you are in the student or a teacher or whether you are an industry person. So the recent advancement which will fill the major gaps in us update ourselves. In this particular CAD, the recent advancements that can be used for the academics or your examination purpose for your knowledge gain is AlphaFold. It is really a game changer. So in the de novo design, it requires high quality protein structures. That is the reason previously we were only getting the chemical structure by the crystallography. Now, because of this AlphaFold's high confidence structures, docking station, our de novo design opening the thousands of the undruggable targets also, which are able to predict, and we are going to get near to the same as that of the structure what we are predicting with crystallography. With AlphaFold, we are able to get the similar pocket, and we are going to go ahead. Similarly, this basic research of the AlphaFold of a part of Google that is revolutionized, and this has got a Nobel Prize also. The software what we are having advancement is a Pandaomics. For the target identification, so they are performed sophisticated gene pathway scoring and found the relevant targets via deep feature synthesis, casualty inference, de novo pathway construction. It means it will find the problem, it will give the drug, it will give the synthesis, and it will give you a molecule, Pandaomics. Of course, we have already mentioned in the previous slide, chemistry 4.2, the de novo generative engine. It is to design the specific molecular structure which reward and punish the generated molecule based on how well they meet the necessary condition. Both in there, it will fail early and pass early, including the hitting the target and metabolic stability and penetrability. So these are all the recent advancements. In the tomorrow class, I will give you one assignment. All of you be present. Tell your remaining people also to present, because I'm going to give you an assignment in the tomorrow class. So in the afternoon, we are having a remedial class at 2:10. So you are required to join. Now I will be opening the floor for the questions.